Hi, we're going to continue today with the fourth of our chemistry lectures. In part three, the discussion was of a molecule water, which is a polar covalent molecule, which gives water all of its special properties. Now we're going to switch to carbon molecules carbon can form these large biological molecules. The categories are carbohydrates, lipids, proteins, nucleic acids. So those are all important in living cells. So we're going to talk a little bit first just about carbon, and then we'll go through each of those four classes of biological molecules. So we talked about forming chemical bonds, and the formation of chemical bonds is actually what a chemical reaction is. So that when you combine when you combine hydrogen and oxygen to form water, that's a chemical reaction. And the reactants are what you start with. So in this case, the reactants are hydrogen and water. Remember, hydrogen and oxygen only exist as H2 and O2. So they would combine to form the product, which is water. And these are balanced chemically. We're not going to do balanced equations here. If you take chemistry, we'll be doing that then. But I just wanted to show you that what you start with is the reactants. What you end with is the products in a chemical reaction. So here you're taking two substances, combining them to form a different substance. There's something else I wanted to say. You have to keep track. You can't create nor destroy matter. So when you're balancing these, you have to end up with the same number of atoms that you started with. So that would be a chemical reaction. So what we're going to talk about next are carbon-based molecules that are called organic molecules. And in the sciences, organic means something a little bit different than what it does if you go to Sprouts or someplace. There, organic means food that's grown without pesticides and artificial fertilizers. But in chemistry, organic means molecules that are carbon-based, so that contain carbon and hydrogen bound to one another. They can be very diverse, but there are four categories of them that are very important in organisms, and those are carbohydrates, lipids, which are also fats, known as fats, proteins, and nucleic acids, which is DNA, RNA, and some other molecules. In our chemistry discussion, we're going to talk in detail about these three. I'm not going to do nucleic acids right now. A little bit later in the semester, we'll do genetics, and we'll talk in detail then about DNA and RNA. But they are of the class of organic molecules. These are also sometimes called biomolecules because they're important in biological organism. Those terms basically mean the same thing. So organic here means carbon-based, not have anything to do with how it's grown or fertilizers and pesticides. So the carbon atom, I didn't want this to do it this way, is, if you remember, it has the atomic number of carbon is 6, so it has 6 electrons. So it has 2 in the first energy level, 4 in the second energy level. So in order to have a filled second level, it needs 8 electrons. So it needs 4 more. And so what it does is that it can form covalent bonds with 4 other atoms to get those 4 more electrons. And because carbon can make these 4 bonds, it kind of gives it a lot of diversity as to the types of molecules it can form. It can also, one of those bonds or more of them can be with another carbon atom. So you can get long chains of carbons bonded together. To give you an example, like octane, which you probably heard of when you pump gas, they talk about it has a high octane level. Octane means it's a hydrocarbon with 8 carbons. So it's 8 carbons bound together. I think I have a picture on the next one. So this is octane. It's just carbons and hydrogens. And remember, carbon needs to form 4 covalent bonds. So if you look at every carbon here, it's got 4 bonds. In the middle, it's a bond with 2 other carbons and then 2 hydrogens. They can even form ring structures where the carbons are connected to one another and can form a ring. We'll look at glucose later. Glucoses usually exist as a ring structure. So these are just 2 little examples of some of the diversity in carbon molecules. So again, carbohydrates, lipids, proteins, and nucleic acids, because they can get these long chains of carbons, can be very large molecules. So sometimes they're called macromolecules, macro meaning big, because they're so big. And the macromolecules are also sometimes called polymers. And a polymer means it has many parts, and the many parts are formed by joining together smaller parts, which are called monomers. So you can join monomers together to make polymers. And all of these are formed, carbohydrates, lipids, proteins, nucleic acids are formed by joining monomers to make polymers. So we're going to go through that in a little bit more detail, since these molecules are so important to biological cells. These are just a picture. You tend to think of carbohydrates, lipids, and proteins as food, but it's actually what we're primarily composed of. So even though it is what we eat, it's also, we're eating at one time, we're living things. So that's also what our body is primarily composed of, along with water. So to make a polymer, you have to combine monomers. So you can have one monomer, oops, let me go back to, join with another monomer and another monomer to make a polymer where those are hooked together. Okay, so to join these monomers, this is a chemical reaction to form these bonds, hooking the monomers together. And joining monomers to form polymers uses a type of reaction called a dehydration synthesis. If you go the other way, suppose we started with the polymer and wanted to go back to the monomers, we would have to break all those bonds, and that's called hydrolysis. The lysis part of the word there means to break apart. So that's using water to break apart. So those 2 reactions, dehydration synthesis and hydrolysis, are common reactions in making all of our biological molecules and making carbohydrates, fats, proteins, and in breaking them down. So this is just another example, a little drawn better than what I drew for you. So say we have, this is one monomer, and this is another monomer. And we wanted to join them together to start making our polymer. So dehydration synthesis, as you can kind of get from the name, dehydration is removal of water. So what you do is you take a hydrogen from one monomer and an OH from another monomer, that forms your water. And now this bond is now going to be with the monomer. So you've linked them together by removing the water and joining them together. So that's dehydration synthesis. If you wanted to do the opposite, suppose you had the polymer, you wanted to go back to your monomers, then that's hydrolysis. And you're going to add water back, you're going to break that bond and go back to your original monomers. So we do that back and forth all the time. When you're breaking down your food, you're doing hydrolysis. When you're making proteins in your muscle cells, you're doing dehydration synthesis. So those are common pathways used in handling all of our biological models. So what we're going to do next is go through those three major classes of biological molecules: carbohydrates, lipids, and proteins. We're going to talk about their structure, what their roles are in the body. And again, the fourth one, nucleic acids, we're going to do a little bit later in the semester. So we're going to start with carbohydrates. And carbohydrates, if you can kind of tell by the word, the slides switched, and when I'm recording, I can't go backwards. So carbohydrates, as you can kind of get from the name, they are made of carbon, and hydrate is water, so they're composed of carbon, hydrogen, and oxygen. And when you say like carbs are like on the bad side of diets right now. Everybody's trying to cut out carbs, and you tend to think of things like breads and pasta as being carbohydrates, and they have other things too. So most foods are not just one type of molecule, but a combination of molecules. But bread and pasta happen to be high in carbohydrates. So carbohydrates can be formed as monomers and polymers. And the monomers are called monosaccharides. Saccharide, I think, is Greek for sweet, so it's kind of like monosweet. And monosaccharides are the monomers for carbohydrates. So it would be just a single sugar molecule. Sugar is another somewhat misused word, because when we say sugar, lots of times we think of table sugar, which is actually sucrose. It's actually not a monosaccharide. And we say blood sugar, but blood sugar is actually glucose, which is a monosaccharide. So sometimes sugar is used broadly, and it's not as well defined. So here we're going to talk about monosaccharides. One that you're probably familiar with is glucose, but there's a couple other ones that we'll talk about. And then you can join two monosaccharides together by a dehydration synthesis to form a disaccharide. So that's two sugars. Lactose and sucrose are examples of that. And we'll talk more about examples. The third type of carbohydrate are polysaccharides, and they're big polymers of monosaccharides. So starch and glycogen are examples of those, and they can be hundreds or thousands of usually glucose molecules joined together. So with carbohydrates, we can have monosaccharides. The three primary ones that we'll talk about are glucose, fructose, and galactose. Glucose being the primary energy source in our cells. Fructose is found in fruits. It's the sweetest of the monosaccharides, so anything that has fructose in it tastes sweeter. And galactose is found in dairy. So maltose is two glucoses, and we're going to go through this again. Lactose is one glucose plus one galactose, and sucrose is a glucose plus a fructose. So this is two glucose, glucose plus galactose, and then this one is glucose plus fructose. And then the polysaccharides are all polymers of glucose, so there are hundreds of glucoses joined together. So glucose is a ringed carbohydrate. Its structure is C6H12O6. That's actually also the chemical formula for fructose and galactose. They're all C6H12O6, but the reason they're different is the arrangement of the atoms are different in glucose, fructose, and galactose. But you'll see it represented like this, like this. There's like, these are abbreviated forms. You don't need to know how to draw these, but you should recognize when you see this ring structure that it's glucose. So again, the monosaccharides that are important physiologically that we'll be talking about are glucose, fructose, and galactose. And this shows they're all C6H12O6. So if you were to count the carbons, oxygens, and hydrogens on each of these, you'll see there's 6 carbons, 12 hydrogens, 6 oxygens, but you can see that they're arranged a little bit differently. So that's why you have 3 different molecules. So this is a single monomer or monosaccharide. These taste the sweetest. They're a primary source of energy for cells. So glucose is the sugar in our blood. It's the only thing, even if you eat dairy, it gets converted to glucose. There's no galactose in your blood or fructose in your blood. Excuse me. And as I mentioned, fructose is primarily in fruit and galactose in dairy. So as I mentioned, you can join 2 monosaccharides together and make a disaccharide. And that's done by a dehydration synthesis reaction. So a disaccharide is 2 monosaccharides. The 3 you're probably most familiar with is lactose, which is in dairy. And it's composed of 1 galactose and 1 glucose. Sucrose is in table sugar, and it's glucose plus fructose. And then maltose is 2 molecules of glucose. And that's formed sometimes when if you had like a chocolate malt or a malted milk ball, you would have maltose. And it's also when you like toast things, some of the starch breaks down to maltose. So those are the 3 primary disaccharides that are in our food that you're probably familiar with. So again, when you're making a disaccharide, you use a dehydration synthesis. So here is the example if you were making maltose, which is 2 glucoses. So you have 1 glucose and another glucose. And remember, it's a dehydration reaction. So you're going to take the OH from this glucose and an H from that glucose and make water. And then this oxygen is now going to bond with the other glucose. So now the 2 glucoses are not separate molecules, but are joined together to form 1 molecule of maltose. If you were going the other way, suppose you ate something with maltose in, we only can absorb monosaccharides from our small intestine. So you would have to now break that bond through hydrolysis by adding the water back and forming the 2 glucoses again, and then they could be absorbed into your blood from your small intestine. Likewise, if you were making sucrose, you would take a glucose and a fructose and do a dehydration reaction to form the sucrose, which is the disaccharide. If you ate sucrose, you would have to then break it down in your small intestine using hydrolysis to break that bond and go back to glucose and fructose. So this is just showing that same reaction then again for sucrose. So sucrose is a disaccharide. Glucose and fructose are monosaccharides. So the dehydration, you remove the OH and an H to form the water. And then the glucose bonds with the sucrose to form the disaccharide. The next level up, so we talked about monosaccharides, disaccharides. The next level of organization are the polysaccharides, and poly means many. So there are many monosaccharides joined together. The ones that are important to us physiologically are polymers of glucose. So they're glucose plus glucose plus glucose, etc. I'm going to talk two polysaccharides that are very important because they're primary sources of energy. One is starch. So starch is a polysaccharide found in plants. Shoot, I went back. As I said, I can't go backwards, but this has the same picture. So starch is a polysaccharide found in plants. And it is hundreds of glucoses attached to one another. So here's a glucose attached to a glucose, to a glucose, to a glucose, to a glucose. And that can go on for hundreds or thousands of glucose. So it's a very big molecule. And so that's why when you eat a starchy food compared to one with monosaccharides or disaccharides, it takes longer to digest it because you have to break all those bonds. Whereas if you're eating sugar, you only have one bond to break to digest it. Whereas if you're eating starch, you could have hundreds of bonds to break. So the starch, the reason the plant has starch is plants primarily store their extra energy as carbohydrates, whereas we primarily store our extra energy as fat. So a potato is really a storage of carbohydrates, of glucose in the plant. So when we eat the wheat or the potato or whatever starchy food we're eating that plant cells stores of glucose. As an animal, we don't store a lot of carbohydrate, but animals do store a little bit. And the storage form of carbohydrate in animals is called glycogen. It's really almost identical to starch in that it's also a polymer of glucose. And you get these long glucose molecules. We store a little bit of glycogen in our muscles to have kind of a ready supply of energy available in your muscle if you start using it. And the other place we store some glycogen is in our liver. And in the liver, it serves as a readily available source of glucose. If your blood glucose level starts to drop, you can break down your liver glycogen into glucose and release it into the blood and bring your blood glucose level back up again. But most of our extra calories, even if we eat extra calories in the form of glucose, we don't store a lot of glucose. We convert it to fat and store it as fat. The third polysaccharide that's important but does not supply energy is cellulose. And cellulose is found in plants. And in plants, it isn't a source of energy, but it's the plant cell wall. Plants don't have skeletons like we do to give us support. So plant cells are surrounded by a cell wall, which is made up of cellulose. Cellulose is also a polysaccharide, and it's a polymer of glucose. So it's hundreds or thousands of glucoses together, kind of like starch is. But the problem is the bond holding the glucoses together is different, and we don't have the ability to break that bond. So if we eat cellulose, we don't get any energy from it because we can't break it down into glucose to absorb it into our blood. So cellulose to us is a major component of fiber in our diet. Fiber is parts of your diet that you are unable to digest and they pass through your digestive system and get excreted in the stool. So they're important because they add bulk to your diet, to your stool, and help retain water for a better consistency of stools. But we cannot get any energy from cellulose because we cannot break it down into glucose. So for us, it's a component of fiber. In plants, it's a structural component. And then starch and glycogen are the components or the polysaccharides that we can use for energy. So again, cellulose, this is just a little bit more about cellulose. So it's the major component of plant cell walls. So that's what paper is, because paper and wood are the remains of the plant cells. It's the plant cell wall. So even if you're hungry, you can't eat paper to get, even though it's a polymer of glucose, but since you can't break it down, you can't get any energy from it. So that's the end of carbohydrates. The next PowerPoint and the last chemistry one is going to talk about lipids and proteins.